All right, by now I think all of you should be expert in how we form an action potential as a result of the summation of stimuli that are brought to a neuron. Remember that neurons receive connections from potentially thousands of other neurons, so the summation of the stimuli is very complicated. Many of the stimuli are depolarizing. Many of them may be hyperpolarizing. So it is the summation of those stimuli brought to the trigger zone located in the axon hillock that determines whether or not our local potential becomes an action potential. Well, if it does become an action potential, then what we do is open sodium channels in the membrane of the axon. Remember that axons can run for very long distances, perhaps as many as 2 or 3 feet. Some axons are covered by a phospholipid protein combination that we call myelin. Myelin is made or is contributed by oligodendrocytes in the central nervous system. The same myelin is contributed by Schwann cells in the peripheral nervous system. So here in the illustration, we have a myelinated axon, and down below we have a non-myelinated axon. So what this is a fatty substance which begins to form in the fetal months, is most accomplished in the first 2 years of life, but actually can continue throughout life. So most of the myelination of axons takes place by the time a person is 2 years of age. I think I mentioned earlier that this is why it is necessary for babies to have included in their diet foods that are rich in fats, because myelin is largely a fatty material and it is necessary for proper nervous system activity. Notice that the myelin is compartmentalized. There are areas where there is myelin, those are called internodes. And then there are the nodes of Ranvier, small areas between the individual compartments. When we have a myelinated axon consisting of internodes and nodes of Ranvier, then what happens is a thing called saltatory conduction. The influx of sodium takes place at the nodes of Ranvier. So the depolarization jumps from one node to the next. I would compare it, let's just go ahead and compare the unmyelinated depolarization to the myelinated. The way that I would illustrate this is the unmyelinated axon depolarizes sequentially like a slow burning fuse. In the case of the myelinated axon, having the sodium gates positioned at the nodes of Ranvier, then the depolarization takes place in jumps, like a kangaroo would jump from one location to the next, to the next, to the next. Well, what that translates into is a great difference in the speed of transmission. An unmyelinated axon may be able to transmit an action potential at, well, maybe 5 or 10 meters per second. Well, that may sound fast, but wait till you hear this. If the axon is myelinated, then the speed of transmission of the action potential may be as much as 150 meters per second. So then we're talking about the presence of myelin directly affecting the speed of transmission of the action potential. So we have some axons in the body which are myelinated, those are the ones where we need the rapid transmission of action potentials. On the other hand, we have many axons in the body that are not myelinated because it is not necessary that the action potential be transmitted that rapidly. Now, we can see the importance of myelin when it is damaged or when it is not there. So we do have certain conditions that are demyelinating conditions or demyelinating diseases. Two of the most commonly referenced demyelinating diseases are multiple sclerosis and Guillain-Barré syndrome. Both of these are abnormalities of the immune system, so they're classified as autoimmune diseases. For reasons unknown, the body's immune system begins to attack and destroy the myelin that's present on the myelinated axons. So there are a great number of problems, obviously, that are going to go with. Some of those are motor problems. Some of those are cognitive problems. Some of those are visual problems. So when the myelin sheath is lost due to the advancing of the autoimmune diseases, then the person becomes more and more and more incapacitated. So myelin has a purpose that it serves, and we see the importance of that purpose, especially in people who are losing their myelin. Now, there are also other causes of demyelination. One of the most commonly referenced is exposure to certain chemicals, chemicals that are called organophosphates, particularly present in various insecticides, such as the one I told you about, malathion. So anyway, exposure to certain chemicals, certain viral infections, and then the autoimmune problems that can result in demyelination and the complications that go with that. So the difference between regular conduction of an action potential and saltatory conduction that is offered by myelinated axons. Next. Well, it was originally thought that one neuron actually physically contacted the next neuron, so the action potential could be transmitted directly in that way. I think I mentioned to you that it was Ramon Cajal and his buddy Camillo Golgi in the late 1800s, early 1900s, who looked at this under a microscope and said, hey, looky here, these cells do not even touch each other. There is a space between them. Well, we call that space the synaptic cleft. So the question is, how does the communication go from one cell to the next when there is a gap in between? Well, the answer to that question is that the chemicals that are released from one bind to the next one and in whatever way relay the action potential or communication from one cell to the next. So those are the neurotransmitters. They are chemicals. We know of more than 100 chemicals that are used as neurotransmitters. We have already talked quite a bit about acetylcholine, but we have several other neurotransmitters that we'll talk about in this particular presentation. So the reason this becomes important is because we are focusing on one of the body's principal mechanisms of cell-to-cell communication. If that communication bridge is a chemical, then potentially the communication bridge can be altered, either increased or decreased or otherwise changed by the application of other chemicals, which we would simply refer to as medicines. Well, that's one of the biggest areas of pharmacology these days, is using chemicals to change the activity of neurotransmitters, therefore changing the communication of messages, therefore hopefully improving the health of the person who receives the chemotherapy. Okay, so let's look at this a little more closely here and talk about some of the major neurotransmitters individually. Well, at first glance, this illustration might appear to be too busy and too complicated and too detailed. Actually, it isn't. It's simply a repeat of things that I have already said several times over. So all we're talking about here is the release of a neurotransmitter into the synaptic cleft and the binding of the neurotransmitter to the receptors on the postsynaptic neuron or on the target tissue. Okay, so when you go through this, you can see that it only shows things that I have already talked about. The reason I wanted to look at it up close here is because of the reference here made to the enzyme monoamine oxidase. So number 8 in this illustration, the reference to MAO. Earlier, I had said something to you about the enzyme acetylcholinesterase. When acetylcholine is the neurotransmitter, which it is in the case of stimulating skeletal muscle, then as soon as the acetylcholine has exerted its effect, it has to be taken back up into the presynaptic neuron, or it has to be absorbed by astrocytes, or it has to be broken down and degraded by the enzyme acetylcholinesterase. So neurotransmitters have a brief moment in time in which they can exert their effect, and if that is not terminated immediately, then the neurotransmitter will continue to stimulate the postsynaptic neuron or the target tissue. So in the case of acetylcholinesterase, the example I gave you was the malathion that I was using to kill bugs on my fruit trees. The acetylcholinesterase degrades the acetylcholine so that the signal stops because the neurotransmitter is degraded by the enzyme. In this illustration, the enzyme being referenced is monoamine oxidase. This is the enzyme that degrades neurotransmitters that we call monoamines. The monoamines include epinephrine, norepinephrine, dopamine, serotonin, and histamine. I will talk about those later. What I want to talk about now is the action at the synapse insofar as this enzyme is involved. It was observed that people who had endogenous depression, that is, depression for which there is no environmental explanation. The person is chronically depressed and sad and pessimistic, but there's no explanation for why. It's not like there was any tragedy that took place. They weren't fired from their job. They weren't thrown in jail. They didn't file bankruptcy. Their wife didn't leave and take up with another person. So there wasn't any explanation for why these people were chronically depressed. Well, it was observed by experimentation that if this enzyme, monoamine oxidase, which degrades those neurotransmitters, epinephrine, norepinephrine, dopamine, serotonin, and histamine, if we inhibit the release of that, not just the release or the activity of monoamine oxidase, then some of those people would experience improvements in their mental outlook on life. So it was one of the first antidepressants ever offered to the world. Well, the problems that go with that is that we have 5 different neurotransmitters that are monoamines. Monoamine oxidase will degrade all 5 of them. So you not only get the desirable outcome, the person's mood or the person's outlook on life may be improved because the monoamine oxidase affects all of the monoamine neurotransmitters. You get a list of side effects that is simply unbelievable by taking monoamine oxidase inhibitor medications. Look those up if you will. The person who's on MAO inhibitor therapy, there are certain fish they can't eat. They can't drink red wine. There are a lot of items that they simply cannot include in their diet, and they certainly cannot mix the MAO inhibitor with other medications. So, because the enzyme monoamine oxidase degrades 5 different neurotransmitters, then you get not only the desired effect, you get the possibility of a whole lot of undesirable effects, some of which are just plain fatal. So this is the trick in manipulating action at the synapse. You have to find a way to get the desired effect while minimizing the undesired effects. Well, it was later found that of the 5 monoamine neurotransmitters that we want to target, serotonin is really the one that would be most useful. So we developed the selective serotonin reuptake inhibitor, which I mentioned earlier. Prozac and Paxil are 2 of those antidepressant medications. So they don't affect the other 4 neurotransmitters, which are monoamines. They affect only the reuptake of serotonin. So this is the pharmacology of the day, finding ways to manipulate activities at the neuron to neuron junction and hoping that the change that takes place actually benefits the person more than it harms the person. Okay, next slide, please. Well, there are more than 100 chemical compounds that have been identified as being used as neurotransmitters. Many of those chemical compounds have other uses in the body also, but they are in some circumstances the neurotransmitter involved between neurons and another neuron or a neuron and a target tissue. We have already talked about acetylcholine. This is the neurotransmitter uh, for the purpose of stimulating to contract skeletal muscle. Acetylcholine is also one of the neurotransmitters that we'll talk about in the autonomic nervous system. It is in a chemical class by itself. Acetylcholine is very unique. It is simply an organic molecule called choline, which contains nitrogen plus vinegar. So it's vinegar plus choline. So we classify it as an ester, but as a neurotransmitter, it kind of sits in a class by itself. So you know about the stimulatory effect of acetylcholine on skeletal muscle. When we talk about acetylcholine and its application in the autonomic nervous system, what we will find is that it has inhibitory effects at other tissues. A good example of that is acetylcholine released by the vagus nerve onto the heart It doesn't stimulate the heart, it slows the heart down. So it has an inhibitory effect on cardiac muscle tissue, while at the same time it has an excitatory effect on skeletal muscle tissue. So that is true of all of the neurotransmitters. So it isn't the neurotransmitter so much as it is the receptor that determines the response of the target tissue. Okay, we have several amino acids that are used as neurotransmitters. Glycine is one that is commonly referred to, present in the spinal cord. This is one of those neurotransmitters which will prompt the opening of chloride channels. So it will create an inhibitory postsynaptic potential in the spinal cord. So glycine is simply an amino acid. When it is used as a neurotransmitter, it opens chloride channels in the postsynaptic neuron. Therefore, we get an inhibitory postsynaptic potential. Gamma-aminobutyric acid, or GABA, is another amino acid which, when it binds to the receptor, it will open chloride channels, creating an inhibitory postsynaptic potential. The difference between the glycine and the GABA is that GABA is an inhibitor in the brain. Glycine is primarily an inhibitor in the spinal cord. Glutamic acid, this is the most commonly used amino acid neurotransmitter in the brain. It does create an excitatory effect, so it is an excitatory postsynaptic potential created by the opening of sodium channels. So glutamic acid is very much involved in such brain functions as memory and learning. Aspartate, or aspartic acid, also is an excitatory neurotransmitter. The difference between it and glutamic acid is location. So the aspartate is primarily used in the spinal cord. Glutamic acid is primarily used in the brain. Now we get to the monoamines that I was talking about on the previous slide. The monoamines are amino acids that do not have a carboxyl group. So it is part of an amino acid, not the whole thing as we had in the other category. So the monoamines, norepinephrine, epinephrine, and dopamine, those three are made from the amino acid tyrosine. So all three of those are called together catecholamines. They are synthesized from the amino acid tyrosine. In the case of serotonin, serotonin is made from the amino acid tryptophan, which is the same amino acid from which we make the hormone melatonin. So melatonin and serotonin are cousin compounds. Both of them are made from tryptophan, and the two chemicals are actually interchangeable. So then the last one, histamine, that is made from the amino acid histidine. Okay, let's go back up to the top there to norepinephrine. In the autonomic nervous system, this is the stimulatory neurotransmitter used in the sympathetic nervous system. You should know what the sympathetic response of anything is. The sympathetic response is the same response that you would get from the hormone epinephrine. Well, all of you know what the hormone epinephrine does. Epinephrine will increase the heart rate, it increases the blood pressure, it increases the volume of air that comes in and goes out, it will dilate the pupils, and it will dry up the mouth. So it's the fight or flight emergency type of hormone. Well, we're not talking about hormones, we're talking about neurotransmitters. Well, these neurotransmitters have the same effect as the hormone epinephrine, except neurotransmitter activity is immediate. Hormonal activity is slow and long lasting. Hormones go into the blood and they circulate through the body, so they have a slow onset and a long-term effect. When used as a neurotransmitter, the response is immediate. So in the case of norepinephrine and epinephrine, we have the same effect. If it is used as a neurotransmitter, that effect is immediate. If it's used as a hormone, that effect is slower onset but longer lasting. The other catecholamine, or neurotransmitter, which is made from tyrosine, is dopamine. This is one of the most important neurotransmitters in the body. The obvious need for dopamine is seen in people who don't have it. So it is involved in skeletal muscle control, and we see how important that is in Parkinson's disease. A person doesn't have much dopamine. The cells in the brain called the substantia nigra, which make dopamine for reasons unknown, they stop making it. So the person is dopamine deficient. Now, here's an interesting thing. We have skeletal muscle control and also mood. Well, it is well known that dopamine is a good mood neurotransmitter. Uh, People feel better if their dopamine level is what it should be. Now, this is an interesting thing that you should always be mindful of. We're talking about a body that has basically an infinite number of chemicals. Well, these are some of those chemicals. Many of them exist on a seesaw with other chemicals. So you know what a seesaw is. You got one person on one side and another person on the other. Well, these neurotransmitters oftentimes have a seesaw type of relationship with other neurotransmitters. The case of dopamine and acetylcholine sitting on a seesaw is oftentimes used as a good illustration of this. If the dopamine level is low, then that means the acetylcholine level will be high. They are on the same seesaw. So in the case of high acetylcholine levels or low dopamine levels, then you have Parkinsonian type symptoms or skeletal muscle control problems. The Parkinson's patient has tremors, they have difficulty initiating movements, and once they get started moving, they have a difficult time stopping. So how would we treat that? Well, we could increase the dopamine by the application of L-DOPA. Alternatively, we can decrease the level of acetylcholine. Since they are on the same seesaw, we would get the same effect. So an anticholinergic drug, something that reduces acetylcholine levels, would balance the seesaw in the same way that giving a dopaminergic drug, something that increases dopamine levels. So anticholinergics are useful for treating Parkinson's disease, just like dopaminergics, such as L-DOPA, are useful. Now we have then the other complication here. We have mood. This is the good mood hormone, a good mood neurotransmitter, dopamine. There, there are moods that become just plain crazy. So let's look at our seesaw from the other extreme. the dopamine levels are too high and the acetylcholine level is consequently too low. What you have is a person who demonstrates the symptoms of psychosis. Schizophrenia is believed to be due to excessively high levels of dopamine relative to the level of acetylcholine. So, how do we treat schizophrenia or psychosis? Well, we give anti-dopaminergic drugs such as thorazine or stelazine, medications that reduce dopamine levels. Have to be careful. If you reduce the dopamine too much, the person begins to show symptoms of Parkinson's disease. If you don't reduce the dopamine enough, then that good mood hormone becomes kind of a manic. I said hormone, I'm talking about neurotransmitters here, a manic type state, such as you would see in a psychotic person. So don't forget that these chemicals do not act independently. They are on seesaw type relationships with each other. And if you affect one, you can't help but affect the other. That's why these types of medications come with so many side effects. In the case of serotonin, since it is the cousin of the hormone melatonin, and we know that the release of melatonin oftentimes is the initiation of our sleep-wake cycle. We take melatonin to help to get to sleep. Serotonin and melatonin are interchangeable. Both of them are made from tryptophan, so we have obvious connection there with the sleep-wake cycle. Serotonin also is involved in mood regulation. Consequently, it is targeted for treatment of depression and sometimes obsessive compulsive tendencies, sometimes anxieties. So medications that increase serotonin activity at the synaptic cleft are commonly prescribed these days to moderate a person's outlook on life or their behavior in a social setting, etc. Serotonin also is involved in the clotting of blood. We'll talk about that when we get to the blood chapter. In the case of histamine, this is our monoamine, which is made from the amino acid histidine. Histamine is the body's favorite vasodilator. Anytime histamine is released, then the capillaries dilate. When you get dilation of capillaries, then you get increased blood supply to the area. Well, there are some problems that can go with that. We call the increased flow of blood to an area, we call that inflammation. Well, inflammation can hurt. It can also itch. It can also damage tissues. So, in the case of histamine, you're probably most familiar with it uh, in that we try to block it because we have discomfort or we have pain or we have watery eyes or things of that sort due to the release of histamine. So, we take antihistamines. Okay, the next chemical class there, the neuropeptides. Well, these also are amino acids. They just happen to be bound to each other. That's what we call a peptide, amino acids bound to amino acids. So we have neurotransmitters such as substance P, which mediates the transmission of pain signals. Well, notice that we also have a neurotransmitter that will inhibit that. Those are called enkephalins. Then we have the neurotransmitters called endorphins. also called endogenous opioids, reducing the perception of pain and fatigue. So these are naturally produced by the body tissues. Some people have reported, that if you stress the body enough, the body will release this beta endorphin and you suddenly begin to feel good. So they call that a runner's high. The release of the endorphin suddenly diminishes the person's perception of pain and fatigue, and they'll tell you they actually feel good. Well, I have never gotten to that place, so I'll just have to believe it, when somebody else says it. The last one listed here is cholecystokinin. Cholecystokinin is made in the small intestine. Its primary function is to stimulate the gallbladder to release bile. Well, that's not what we're talking about here. We're talking about the neurotransmitter effect of cholecystokinin, which binds to cells in the arcuate nucleus of the hypothalamus, and it inhibits the appetite. So when the small intestine says, hey, 30 tacos is enough, buddy, then it releases cholecystokinin. The cholecystokinin binds to the arcuate nucleus of the hypothalamus, which shuts down the appetite. Okay, so those are some of the neurotransmitters. Like I said, there are more than 100 of them, and and they all have a seesaw type of relationship with each other. And so altering the level of one can't help but alter the level of others as well. Next. Well, nothing in human physiology is quite as simple as it might appear at first. And certainly in the nervous system, we see more complexity than we ever imagined. So, in a simplified design, an action potential is just sent from one neuron to the next, and a certain response is generated. Well, it isn't nearly that simple. We have discovered that there are things called neuromodulators which can affect the neurotransmitter release or the binding of the neurotransmitter or the response of the postsynaptic cell to the neurotransmitter. So I have drawn some funky little lines here and stuff. So let me see if I can explain a little bit about what I was trying to get across to you. So you recognize that we have a presynaptic cell and we have a postsynaptic cell. Well, in this case, I have drawn a little blue line from the postsynaptic cell, and this is in the upper part of the image here. The little blue line going back to the presynaptic cell, which is saying, hit me again, Clyde. So this is communication going from the postsynaptic cell to the presynaptic. What we've talked about so far is communication going from presynaptic to postsynaptic. Well, it turns out that the postsynaptic cell may have some say-so over what the presynaptic cell does. So in this case, the postsynaptic cell likes what the presynaptic cell is doing and sends back a message saying, do that again. I like it. So hit me again, Clyde. Or we can have outside of this two neuron design, also in the upper part of the illustration here, other tissues of the body releasing neuromodulators that affect what happens at the synaptic cleft. So here I have a heart being an example of an organ releasing a paracrine or a neuromodulator chemical that affects what the presynaptic neuron does. So the heart may be saying to that neuron, what you're doing is a good thing for me, therefore give me more of that. So this is neuromodulation. It's affecting what the two neurons normally do. Uh, as I said, physiology is more complicated than 1 plus 2 equals 3 because there are millions of factors that come into play here. Now down in the bottom part of the illustration, the blue lines I have drawn here, the postsynaptic cell is feeding back to the presynaptic cell, saying, now cut that out. So whatever it is you're doing to stimulate me or to inhibit me, I'm sending back my feedback telling you to cut it out, or I'm telling you don't pay any attention to what the other cells are saying and listen to me. So this is neuromodulation, communication going from the postsynaptic back to the presynaptic cell. Now, the reason I point this out, perhaps you have heard about the original research done on a chemical called sildenafil. Well, you would recognize sildenafil as being the generic name of the medication Viagra. Well, the researchers who were doing this work were experimenting with sildenafil, which is a vasodilator. The actual research was on the coronary arteries. So what the researchers wanted was a medication that would dilate the coronary arteries and therefore allow more blood to go to the heart. Well, what they found out is that the sildenafil not only dilated the coronary arteries, it dilated arteries going to other body parts also. Did I say I'm talking about Viagra here? So you fill in the blank. So the researchers were taken by surprise here. They said, you know, we're trying to improve this guy's blood supply to his heart, and lo and behold, look at that. We have improved the blood supply to another body part, and so the medication is not sold to people with coronary artery problems. It is sold to people who have other problems and has turned out to be one of the most marketable and money-making medications ever made. Well, how did it happen? Well, it turns out that the sildenafil stimulates the postsynaptic neuron to release a neuromodulator, which is simply nitric oxide. When the postsynaptic cell releases the nitric oxide, it feeds back to the presynaptic cell saying, hit me again, Clyde. And so what we get is the desired result of vasodilation because the neuromodulator, nitric oxide, continues the circle of stimulation that is needed for the desired outcome to take place. So that is a good example of a neuromodulator at work. There are actually some very strange chemicals that are used as neuromodulators. One of those is carbon monoxide, which you would normally think of as being deadly poison. Carbon dioxide also is a neuromodulator. Nitric oxide is the one that was focused on in this Viagra research. So that's how neuromodulation works. So again, in the field of pharmacology, where we're trying to change people's physiology by the use of chemicals, then we have to consider uh, the effect of neuromodulators and how they can be manipulated to hopefully arrive at a good outcome for somebody. Well, you know, science is mostly about observation. We observe what goes on and we document what goes on. We try to establish a sequence of events that take place in what is going on. And then we also try to identify certain cause and effect relationships between the things that are going on. So we do a lot of observing. Now, that is different from explaining how or explaining why something goes on. We simply observe and we document and we try to establish relationships as best we can, much of which is purely speculative, some of which can be objectively proven, I guess you would say. But in the areas of how something happens or why something happens, we are pretty much at a loss for offering an explanation for that. So, such is the case with something like learning. What we have are neurons in the brain. Well, we have already established that neurons receive action potentials from other neurons or from sensory receptors. Well, we have a sense of vision, we have a sense of hearing, a sense of taste, sense of smell, sense of touch. Well, we have action potentials that are sent from those sensory receptors. So it is only a wave of depolarization that arrives at the neurons in the brain. The question is, how does the brain remember? Well, we haven't explained that at all. How does it remember? Well, the only thing that we can do is document what we observe. That learning is oftentimes simply a matter of repeated exposure or a repeated attempt at doing something. So we have two types of learning. We learn procedural things such as tying shoes or riding a bicycle, or we learn declarative things such as remembering the capital city of every American state. Uh, Repetition, repeated encounters seem to be the most valuable tool to getting our neurons to learn new things. When you were born, you couldn't walk, but you kept trying, and so you got pretty good at it. So by continually challenging the nervous system to do something, then you learn to do it. When you were born, you couldn't read, but you were exposed to it and exposed to it and exposed to it, challenged by it and challenged by it, and by now you're very good at reading. So, whatever you might take on, learning it is simply a matter of repeating the experience over and over. So, you as a college student taking a class, I have told you many times that I can speak. Well, when I speak, you hear it. Well, that's one thing, but I told you to do more. I told you to take notes. Well, that means you write down what you heard. Well, that's exposure number two. Well, there's more. I told you to draw and label things. And then I told you to say them out loud. Well, that increases the number of times that you expose yourself to it. So you hear me say it. You see what I have written down on the PowerPoint, or if we were in the classroom on the chalkboard. Uh, you write those things down that I say. You draw those things that I have illustrated, and then you speak them aloud. And then I tell you, you need to tell little sister about this, because the more you speak it, the more you learn it. It's my opinion that a person really doesn't know anything unless they can teach it. So all we're doing here is observing things that contribute to learning. Explaining it, well, all that we really know here is that an action potential is initiated at these sensory receptors. It terminates in certain cells of the brain, and if those cells of the brain are exposed to it multiple times, then they begin to recognize it and they begin to have learned it. So learning is a very interesting thing, and it's what you are up against now, trying to learn anatomy and physiology. I want you to learn it. So I recommend to you the things that are essential to learning. So don't just scroll through a PowerPoint. You need to go back and play it again, and you need to write down, and you need to look up things that you're not sure about. Drawing and labeling in anatomy and physiology, those are very effective tools for learning. Certainly, speaking out loud, you need to say these words and you need to tell other people about them. Or if you don't have other people to tell, find a tree and go out and tell it. The more you speak it, the better you're going to learn it. Anyway, so that's what we know about learning. Most of it is just observation. We don't really have an explanation for how the neurons actually do this. Very much the same with memory. We have an action potential that arrives at a certain collection of neurons. The action potential stimulates those neurons. If it doesn't come around again, chances are it will simply be forgotten. So we have short-term memory and we have long-term memory. Short-term memory would be illustrated by I could give you my phone number. Well, you would remember my phone number for about 30 seconds or maybe 60 seconds, long enough to call me. But if you didn't go back and revisit that, then you would forget it. And that's just the way the short-term memory works. It's good for a certain number of seconds, and if it's not recalled and used again, then it is simply erased. Well, going to college, you have plenty of opportunities to have short-term memories. The things that you're exposed to in your classroom work, the hope is that it will become long-term memory, something that is stored inside your head that you can call up and use when you want to or when you need to. Well, we have an area of the brain that is called the hippocampus. The hippocampus seems to be the area of the brain where the decision is made. Should I store this in long-term memory or should I just simply erase it? So the hippocampus kind of processes incoming sensory information and decides whether it should be stored as long-term memory and therefore kept in areas of the brain such as the temporal lobes where we keep our long-term memories. Apparently, the hippocampus determines this while a person is sleeping. So the things that a person is exposed to in the course of a day while the person is sleeping at night, the hippocampus is making decisions about whether this should be erased or whether this should be stored away in long-term memory in the temporal lobes. So what factors play into memory? What things are important to the hippocampus such that we remember something? Well, one thing that we have observed to be most memorable is the shock value of the event. The more shocking it is to a person, the more likely it is to go from the hippocampus to the temporal lobes and be stored there permanently. For instance, people of my generation, we all know where we were. We know what time of day it was. We know what the weather was like. We know what the look on people's faces was like when we heard the news that President Kennedy had been shot. Well, that's because it was a very traumatic, it was a very uh, shocking piece of news. The same thing happened again in 1968 when Martin Luther King was assassinated. Everybody remembers where they were, what they were doing, and what the weather was like that day. So those things are shocking. The attack on the World Trade Center and the Pentagon in 2001, those were shocking things that are stored in long-term memory for everybody. So the shock value is one way to basically guarantee that something will be put in long-term memory. The other thing is simply the repetition. The more something is done, the more the hippocampus considers it to be important and therefore worthy of long-term storage. So this is why repetition. You don't get it right the first time, you do it again. When I try to learn a new song, I cannot play the thing worth a crap. I just, it takes going over it and going over it and going over it and going over it before I'm ready to record it because it takes repetition for me to get all of the the parts in order and to deliver something that is something that resembles music. So memory is established basically on two things: the shock value of it or the number of times it is repeated. So the more times you read a book, the more familiar you're going to be with it. The more times you experience something or try something, the more likely it will be stored in the long-term memory. But even long-term memories can be lost if they are not revisited. So it is necessary to call up long-term memories periodically and to refresh them. Okay, what about this thing called synaptic plasticity? Well, you know that we have synapses. Those are the connections between neurons. Every neuron has the potential to establish connections with an infinite number of other neurons. Learning and memory are the establishing of new synapses. So when we learn something, when we commit something to long-term memory, what we do is build a physical connection between neurons. We establish a new synapse. So that is called synaptic plasticity. Neurons have the ability to make new connections with other neurons lifelong. So we don't increase our number of neurons. The number of neurons that we have at birth is about all we're going to have, but those neurons can continue to make new connections. The more new connections they make, then the better you are at whatever it is you're trying to do. So there are some things that can maximize a person's synaptic potential, and that is to always be learning something new. Now, the danger that you face is this: you're going to graduate from college. Uh-oh, uh-oh, you graduate from college and your brain is no longer stimulated with new information, therefore is no longer challenged to make new synaptic connections. Your life becomes very routine. Day by day, you do the same thing. Nothing new comes along. You know, you got your job and you got your family and you got the routines that you carry out. And so nothing new is taken on, nothing new is learned, no new synapses are made after a person graduates from college unless you intentionally cause it to happen. So the idea here is that once you graduate college, you need to continue to stimulate your nervous system to keep making new synapses. If you stop doing that, if you stop challenging your brain, then it will go into decline. Synapses that are not used are erased. So synaptic plasticity goes both ways. You can make new connections by challenging your nervous system, or if you decide to not challenge your nervous system and you just go into a day by day, everything is the same, you don't learn anything new type of lifestyle, then the connections that you have will be erased. The memories will be lost. The things that you learned to do in college, you will forget. So you have to continue to challenge the nervous system. So I always tell my students when you get out of school, you need to initiate new learning challenges. Learn to play a musical instrument. If you already play one, well, learn to play a new one. Learn to speak a new language that will cause new connections to be made. Get yourself a copy of Moby Dick and memorize the thing. Whatever it takes to keep your brain moving forward. Because if it isn't moving forward, it will move backwards. And so by the time you get to be about 60 years old, you'll start wondering what has happened to my memory? What has happened to my brain? What happened to my ability to ride a bicycle or to dunk a basketball? Whatever, if you don't use it, you lose it. So my little proverb to go with that is this: the electric chair and the rocking chair both accomplish the same outcome. Think about it.